Good afternoon everyone. Today I would like to take a moment. I am Nur Fatima, master's student at University of Arkansas. I work in Dr. Seba's lab. My research topic is comparisons of catalytic efficiency between free enzymes in solution and enzymes bound to engineered mobile enzyme sequestration platform or MESP. Lignocellulose is one of the most abundant biopolymers on earth. It is composed of cellulose, hemicellulose and lignin, which may be further decomposed by yeast in ethanol fermentation. Lignocellulose sources such as corn and sugarcane are the basis for more than 90% of world's ethanol supply. These are the first generation feedstock and has been subject of debate. Hence, the production of ethanol from these sources can impact food supply. An alternative of first generation feedstock is second generation feedstock, which include corn stover, baggies, rice straw, forest waste tree. They are most promising feedstock due to its global abundance and availability. Two steps are involved in the ethanol production. One is pre-treatment step and another is enzyme mediated sugar reduction. However, enzyme mediated reactions are often less efficient due to harsh environmental condition of industries such as high temperature and low pH. To overcome this pitfall, our lab developed a chimeric mobile enzyme sequestration platform or MESP. So, what is MESP? MESP is a fusion protein between one of the archaeal HSP or heat shock protein and coesin from anaerobic bacterium Clostridium thermophilum. Here the gray color represents the HSP beta, which comes from Cephaloba shebate, and the red indicates the coesin domain from Clostridium thermophilum. Blue color represents lignocellulose deconstructing enzyme bind through dockerin, which is showed by green color, to coesin. As it was engineered with HSP beta protein, it is called HSP beta co. Some works have been done with the HSP beta co. To differentiate my work from the previous work, I re-engineered the system and used HSP beta co and HSP alpha from Cephaloba shebate to make it heteromer. When half of the MESP complex can bind with the enzyme by coesin domain and half cannot, because it is predicted that overcrowding may reduce the enzyme efficiency. Another reason behind to use heteromer is that it is more prevalent in nature and hypothesized that it is more stable at high temperature and low pH. Here I chose two enzymes. One was cell K, an exonuclease which cleave at the terminal bonds of the substrate cellulose, and another was xylanase, which is an endonuclease which cleaves the internal bond of the cellulose. All the enzymes have taken from Clostridium thermophilum because it has dockerin which can bind with the coesin domain of HSP beta co. In prior work, some enzymes showed even twofold sugar reduction efficiency with wheat straw and corn stover. The overarching question of my study is, can HSP beta co and HSP alpha heteromer improves catalytic efficiency more than threefold because it is more stable at high temperature and low pH? To determine the sugar reduction, I applied four approaches, which include pre-treated the substrate with 0.1% sulfuric acid for 2 hours at 100 Celsius. Here I try to reduce the concentration of sulfuric acid because it is expensive as well as hazardous to the environment. Second was to purify HSP beta co and HSP alpha to make the heteromeric MESP complex. Here we can see the TM picture of MESP complex with scale bar. In enzyme assay, I applied only enzymes and enzymes bound to MESP in the substrate to compare the sugar reduction. At last, I used Perkin-Johnson colorimetric method to measure the sugar reduction with 96 well plate reader at 690 nanometer wavelength. Here's the result from rice straw. I picked two substrates to see the efficiency of sugar reduction. One was rice straw and another was forestry waste. The result from the rice straw did support the claim, although there was significant improvement but did not approach the absorbance, that is twofold sugar reduction. In each case, three conditions were tested: untreated substrate, which represented by yellow color, free enzyme in solution indicated by blue, and enzymes bound to MESP represented by red. Enzyme efficiency was assessed for two enzymes independently and a cocktail enzyme at 40 degree, 65 degree and 85 degree Celsius. In figure 1, at 40 degrees Celsius, MESP-bound xylanase showed the high sugar reduction, whereas others not. This could be xylanase can break down xylan, which is the main component of hemicellulose rice straw. In figure 2, at 60 degrees centigrade, cocktail enzyme and cocktail enzyme bound with MESP both exhibited similar sugar reduction. A positive synergistic effect has been noticed here. In figure 3, at 85 degrees Celsius, enzyme bound to MESP both singularly and as a multi-enzyme showed significant breakdown of the substrate, although this may have been a product of high temperature sugar caramelization. On the other hand, forest waste was tested with only one enzyme, cell K, and exonuclease, which break down terminal bond. A modest but significant sugar reduction was observed at 40 degree and 65 degrees Celsius, although at 85 degrees Celsius, lowest sugar reduction was observed. From the rice straw, we can see at 85 degrees Celsius sugar reduction was higher, and we hypothesized that it may be the result of caramelization. If it is really caramelization, then why the value of forestry waste was so low at this temperature? More works need to be done to define this, or by using HPLC we can know each component of the mixture. In the present study, these data clearly demonstrate that use of MESP can enhance the sugar reduction efficiency and protect enzyme on acid-pretreated substrate at various temperature. Here is the references that I used to make it. Finally, I would like to thank you, my advisor, Dr. Ruben Michael Sebals, the National Science Foundation for the funding, my team members and the University of Arkansas to contribute of this work. Thank you everyone.